Thank you very much, Wolf and Jay, for giving me the opportunity to present one of our recent data. So you don't be disappointed that I'm not doing genome engineering, but epigenome engineering. I hope that we can catch up. So my group is interested basically in the remodeling of the heart tissue during the process of development and diseases. And today I will only concentrate in the disease part due to the common interest in this round. So basically what happened is that all of us try to find a suitable target, and ideally this target nowadays could be something more precise, so that this is a specific and time specific, and I think this is the beauty of all the progress that we have now, and this is what we are also trying to look for. So we are trying to understand the mechanisms that are driving a normal heart to this heart in order to modify and try to see whether we can reverse the situation. And this is something that we do ideally in this pipeline. So I'm very happy to be here in a medical university that had very good connections to clinical colleagues that can provide us with material from patients, observations for patients that allows us to elaborate in pathophysiological mechanisms which are relevant in order to go to the lab and to, it's working, yeah. To go to the lab and to investigate those mechanisms and to come up with regulatory mechanisms of disease in human and in vivo models, and ideally to perform some therapeutic proof of concept or revert the hypothesis again. So especially what is happening during the course and what is our main interest in is the transcriptional dynamics during this process. And what happened is something which is actually very easy to explain but very complicated to study, which is a loss of homeostatic transcriptional programs and an activation of pathological transcriptional programs. And this is giving a disbalance to the heart that will allow to go to a disease condition. And ideally, we would like to identify those mechanisms and more specifically those that are working in the heart in order to avoid side effects when we decided to elaborate in proof of concept study. So, and this is what we were doing for a long time to identify evolutionary conserved targets and that are heart specific. And this slide actually is a very short slide that summarizes 10 years of work of my group in which we have contributed to the knowledge that the activation of the wing signaling pathway is occurring during the heart failure progression in human, also in animals models, and we have identified also in a specific epigenetic program, and in this case driven by a specific transcription factor, which is TCF7L2, associated to beta-catenin, which is one of the main transducer of the pathway, and many of you may be familiar with the pathway because this is what we use to manipulate our cell cultures. So this is leading to a disease program which is driving the heart to disease condition, and we also know from genetic modifications in animal models that by inhibiting this pathway we can also prevent the progression of disease. The problem is that wing signaling, as you may know, also is very broad and is almost all the organs a very important player. So we decided with my group to go to a more screening approach, and this is a way that I, in the first state, Mauro Jaga had presented how we approach the investigation of certain pathways. So we started with a more hypothesis-driven approach, and now turned to a more screening approach in order to find elements that are targeting or are regulating this pathway specifically in the heart. And by doing that, we have described a protein complex that is regulating the transcriptional machinery of wing signaling, specific in cardiomyocyte. And here I'm showing you only one of the component of this complex, which is a cruppolite factor 15, which have been turning out to be a very strong repressor of the wing signaling in our hands, specifically in cardiomyocyte, in the mouse, but also in human cardiomyocyte. Others have shown that it's playing a role in metabolic homeostasis and is an hypotrophic repressor. So we wanted to focus further in this factor, and we decided to make a step back in our approach. Now we are coming to the omic area. We didn't have that before, at least not in my lab. And having a lot of elements, how to identify the master regulator, we thought, let's see what is the role that CAL15 plays in this picture. And for that, I would like just to introduce you something that many of you know, but just to have it very clear. And this is a transcriptional regulatory network, which is driven by transcriptional factors, which are targeting their own target genes, which are regulons, and these are driven by nodes, which are determined transcriptional networks. And this is an intricate network that it is happening in the reality when you have a remodeling of a tissue in a disease or in development. So we decided to take advantage of single-cell sequencing or single-cell transcriptomic, and we concentrated on the cardiomyocyte because it was our cell of interest at the moment. And we did a series of analysis in the course of compensatory to heart failure progression. And this had been a fantastic establishment of Federico in the lab. And what he did, he took and analyzed the transcriptomic, and then we teamed up with system biology in Heidelberg with Christoph Dietrich that had analyzed these nodes. So what I wanted to have is from cardiomyocyte, from a specific disease condition, which are the factors that are determining all of the regulatory networks that are happening downstream. And I was very happy to identify CAL15 in those networks, and giving us the confidence that this is one of the node regulators, even more upstream as the Wnt signaling, as I thought before. So what Federico did later is he looked for the expression, and doesn't mean that this expression is changed. It only means that it's changing the expression of genes downstream. But in the case of CAL15, it is indeed downregulated, specific in cardiomyocyte, as you can observe upon transoptic constriction, which is a model that we are using for hypertrophy. Not only that, what we also observe is that this is a gene network driven by CAL15, and this is a group of, you can call it a network of score, which is a group of genes which are regulated by CAL15, and with this we determine a score, and this is really upregulated and driving hypertrophy remodeling. So what happened in the disease condition? We and others have shown that it's reduced in cardiomyopathies in failing heart. Other way around, when we induce a knockout animal model, is driving to hypertrophy and loss of function, and the same is true when we do injury heart muscles that we have done at the time in collaboration with Malte and Wolfram, and we can see that they have a loss of contractility, which is more or less what is happening in vivo situation. So we are in the presence of a factor that is downregulated in the disease progression. And the idea that came up at the time, we had two different ways to go. Either a drug design just to block, to induce the complex, or we restore the expression of a very important homeostatic repressor. And this is what we wanted to do when we approach. At the moment that we wanted to do that, as a proof of concept, we thought we need something that allows us to activate endogenous gene transcription in the heart without going to aberrant levels. There was not such a model at the moment. Now we have a couple of models are run in different organs, but still ours was the first one in the heart. And this is a fantastic work of Erik Schoger in the lab, that he was a part of a PhD thesis. And we teamed up with Erik Olsen at that time because he had similar models done with the enzymatic active Cas9. So the model, many of you are familiar with it, is an inactive Cas9 protein associated to VPR, which is an activator. And what happened, this is a first component of the system, which is introduced under the control of myosin heavy chains for cardiomyocyte. And the second component is given by AAV9, which is driving the gRNA, the guide RNA expression. And what happened is this complex will go to a specific locus in a genome that we designed, and we recruit the transcriptional machine and activate gene transcription. So this animal model has been extensively validated and investigated, and we know that it's a good tool for safe enhancement of single and multiple gene expression, and I'm happy that we can use now for our proof of concept, but also many other colleagues around the world that are using this system. So let's see what's happened now with our molecule of interest. So CAL15 was introduced in an AAV9 particles systemically, and we subject the animals to TAC remodeling. So, and here you can appreciate that TD tomato is indicating the expression of Cas9, GFP is indicating the expression of gRNA, which you can see all the components are well expressed, and as you can appreciate here, we have a TAC control which shows the expected hypertrophic remodeling, which is kind of inhibited or blunted when we have our treatment with Cas-15. What happened with the transcriptional of Cas-15? It is restored. As you can observe, we have the typical downregulation that we observe, which is not dramatic, but is sufficient to activate Cas-15 network. And this is restored completely when we use our CRISPR approach. And the same is true for the function. So we have a preserved function, and this goes along also with the reduced hypertrophy of the cardiomyocyte. So now what happened with the cardiomyocyte, which is our target cells? And for that, again, we did single cell sequencing analysis. We isolate the cells by Langendorf technique, and you can appreciate, again, all of the cells are expressing all of the components of the CRISPR system. And I won't go into detail into the analysis. I want to give you some quick data that show how efficient the system is. So here I just marked the cardiomyocyte populations. And as you can appreciate, you can see that in enzyme control, you have the stress marker very low, which are upregulated in the TAC model. And this is a rescue here in the Cas-15 treated animal, which is more similar to the control. And this can be measured as a stress score, and as you can observe, is significantly reduced in the animals with the Cas-15 treatment. So we are in the presence of a cardiomyocyte transcriptional normalization and function with this approach. So this is all good. The next question is this also relevant for the human heart or for the human cardiomyocyte? And because we cannot do human transgenics models, we decided to do that in vitro by using induced pluripotent stem cells. And what Eric did is he introduced a whole system that we have used in the mouse in the iPS cell in a Savior locus and use this for differentiating cardiomyocyte. We can, of course, use for differentiating in any kind of cell types. And then proceed with our proof of concept study. So again, we team up with Malte and Wolfram, and we did engineering human myocardium with those transduced cardiomyocyte. And as you can see here, these are the typical tissues that you have seen along the whole meeting, which are, we call healthy, the one that are cultured and the flexible poles. And I think you can see also the one which are up on isometric construction. So we try to imitate a mechanical stress, and you can see that those tissues look pretty stressed. Not only that, and this was also for us a very important information, it downregulates Cas-15 exactly in the manner that happened in the human heart and in the mouse heart, which gives us an excellent platform to test our therapeutical approach. So we did again the expression of all the components of the CRISPR system and Cas-15. In this case, we did it with lentivirus for the cardiomyocyte, and you can observe here that the restoration of Cas-15 takes place exactly as the in vivo situation. And something which was also very interesting to see, if you see the tissue function, which is measured by the force of contraction, and I'm not expert on that one, but I got a little bit informed, and as you can observe here is the tissue function represented for a control, healthy tissue. And this is what you have in a stress tissue, which is a control. And now if we normalize Cas-15 in those tissues, this function is rescue, which is exactly the same what happened in the mouse. So we were very happy to see this data that are very reproducible in these two species. And now, as a proof of concept, we decided to see what happened with de-differentiation. We have heard a lot about de-differentiation also during this meeting, which is what happened with the cardiomyocyte when they start to fail. And here is just represented by ACTA2 or SMA1. You have also seen this is something that they start to re-express in adult cardiomyocyte when you have an injury, and here it is completely rescue in this case, which gives us the the confidence that the CALF18 transcriptional restoration allows preservation of cardiomyocyte function and transcriptional normalization also in the human setup. So with this, actually, I'm at the end of my talk, and I hope that you could see or share with me the conclusion that CRISPR-based engineering control of transcription is a powerful tool to restore homeostatic gene expression in disease condition, but of course also for other goals, like when you are interested in a gene function, you can also use the system, of course. Specifically here, I show you that allows for enhancement of disease repressor of CALF15 in cardiomyocytes, and that the restoration of this transcriptional network is a suitable target to avoid disease progression by using this approach. And of course, the next step to go, and this is something which is an ongoing approach, is a targeting approach in large animal models. This we are doing in collaboration with Rabea, and I hope to give you next time the final green light to go for further clinical studies. With this, I would like to conclude, and of course to thank my team for this terrific work, and the whole pharmacology for all the support, and our collaboration partners, of course, you for your attention. I will be happy to receive any questions.